Då ska jag presentera nästa programpunkt som är ett program i programmet. Vi kommer ha en liveinspelning av en podcast som heter 25 minuter som leds av Mikael Syding här. Mikael, du får gärna komma upp på scen. Du får gärna berätta, vem är du?
Jag heter Carl Mikael Syding och till vardags så är jag värd för den här podcasten då, 25 minuter. Innan jag började med det så var jag hedgefondförvaltare inom Brummer & Partners-sfären. Jag satt på Futuris i 15 år. Futuris utsågs till European Hedge Fund of the Decade för år 2000 till 2010. Men om ni inte känner till den så är det för att den lades ner 9 månader efter att jag sa upp mig. Det har ingen som helst koppling till varandra utan det var en ren slump. Men i vilket fall som helst, jag sa upp mig där i början på 2014 för att helt enkelt ägna mig åt roligare saker än att förvalta pengar. Och startade bland annat den här podcasten.
Spännande, som vi ska få höra nu. Ja. Hur många har hört den här podcasten i publiken? Ja, det var blygsamt. Så du kommer få en större publik efteråt, men du har en väldigt stor publik idag. Det är bara det att de inte är här. Man kommer också kunna lyssna på den här podcasten via vanliga kanalen efteråt. Så att jag lämnar över ordet till dig och din gäst som är Martin Åsten i form av styrelseordförande för just A1M Pharma. Så att ni kommer inte ge undan A1M Pharma i nästa programpunkt heller. Jag lämnar över ordet till er.
Ja, tackar.
Eller programmet ska jag säga.
Hi, Martin.
How do you do?
Welcome. I'm fine. How do you do?
I'm perfectly fine, thank you. I wonder what you were talking about just then, but that's okay.
All right. We're going to record a live podcast. Please have a seat.
Thank you.
So, Martin, you're the chairman of A1M Pharma.
I have the honour of doing that job. I was elected last year, having served on the board for a short while before then, and having advised the company before that. So I've got deeper and deeper into exactly what A1M Pharma is all about.
Could you tell us a bit about your own personal background? You have quite extensive experience in bringing drugs to, or at least treatments to market.
Yes, indeed. It's a bit of a shock when you look back over the years and realise when they add up 1, 2, 3, 4 decades, it's actually 40 years since I started doing this. And as a result, I've seen generations of products come through from cardiovascular to cancer drugs and now into new fields. And that's really part of why I'm here, because earlier on there was a slide which mentioned one of my alma mater, which was Roche. And I was involved with them as the head of business development for 5 years, bringing that, if you like, or helping to bring that revolution in biologics into the treatment of cancer, which, as a part of my job there, was to try and look forward into the future and say what's possible, what can be done to actually affect and improve the treatments that we bring to patients. That was what I was involved in then. I then went through a period of investing. So I was directly investing into companies from a US investment firm. But now what we see is that we're coming to, if you like, the end of a biologics environment where the first wave has gone through, and now we need new targets. And I think you want to talk to me about what we're doing at A1M, which can address that.
Yeah, we'll get to that. But first, how did you get involved with A1M Pharma? How did you get from Roche and your other assignments to there?
Well, basically, having come through the investment period, that was very interesting, but really wanted to get back into working with companies and working with science, which is where I was able, having actually written a book or two on business development, which was the function I was doing for Hoffmann-La Roche. We became asked to present on master's degree courses at HEC in Paris and Grenoble School of Management, and was asked to do further courses. At which point I was able to meet with Thomas Eriksson, who you've just heard from, as another one of these young people coming through the industry who was looking for advice. that was me.
Well, what's the name of your book? Is it something an ordinary layman can use, or is it for
It depends
academic
what you
use?
want to use it for. If it's putting under a table, it's very good, yes. It's called Business Development for the Pharmaceutical and Biotechnology Industry. And the publisher wanted to have as many words in the title as possible, which you could find on Google.
It sounds pretty straightforward. It sounds like something a normal person could read and understand.
I had a number of people say to me that it explains things in a way which is not so technical that it makes it difficult to use.
Okay, so let's get to A1M Pharma. You have this drug Rosgard, which is a slight change from the A1M Pharma, A1M molecule. How big a change is it? And how difficult is it to make that change?
Well, I'll briefly explaining about proteins. Proteins are made up of amino acids, long chains of amino acids. They take up a particular sequence, and whilst they can be, if you like, chemically identical, you can actually swap one or two of them around, which change some of the characteristics of the molecule, particularly things like stability in solution and the way that it actually holds itself together. So what we've actually done is got the bacteria which produce the protein, not to get too technical about it, to actually alter a couple of the sequences so that we get a more stable drug and a more, I won't say effective because that takes it harder, but
patentable.
Isn't that a good word? Yes. Basically, because we've actually changed the structure, that's our invention. So it is patentable, and we now have a substance of matter patent filed and going through the process, which means that the old accusation of, you're taking something from nature, is no longer true. We've improved upon nature.
I still guess that the reason you are manipulating the protein is not only to make it stable, it's actually to get to a patent, which is, that's good and well. Otherwise, there wouldn't be any business. But could a competing company use the generic endogenous protein for treating this disease?
Well, this was a very interesting turn of events because we have been in conversation through academic links with a company called CSL. And CSL is one of the biggest plasma fractionators. It means they take human blood, which is not used for transfusions, and take the fractions out of that so that the active molecules can be used. Now, they wanted to talk to us because the one thing they couldn't find in the blood was alpha-1 microglobulin, in other words, Rosgard. They couldn't find it because it spends a very short period of time in the blood itself before it's taken into the tissues. So the one thing they didn't have in their deck of cards was A1M.
All right. And how do you actually do it?
What we're able to do is to take a bacterium called E. coli, which is very famous for all sorts of reasons, but it's one of the first bacteria which was used to do recombinant protein manufacture. So its own genome was particularly well sequenced. Everybody found out how that worked. So it's a very stable, well-used platform for producing proteins. Now, other people have used other cells, mammalian cells, and all of these systems have different ways of expressing the proteins. Ours needs to be expressed into what are called inclusion bodies within the bacterium. The reason for that is that the inclusion body then seals off our protein from the inside, the cytosol, this stuff which holds that bacterium together, in a way that means that it doesn't react with those and is therefore capable of being used as a medicine. If alpha-1 microglobulin was just allowed to go to work in any of those cells, we'd never see a product. A bit like what CSL found, which is why we are actually cooperating with them to find out more about that side of things.
Okay. And can you get into a little bit more detail exactly what you're going to do in the different clinical phases?
Absolutely. I mean, when I first came to A1M, the target, the aim was to do a very important thing, which is, as Thomas has told you, the treatment of women who are having a baby whose kidneys are under attack from the baby's blood. Now, for an early stage company, and if you like, I've seen a great many early stage companies, that's quite a long, far target. So what we've looked to do is to find a way to build a stepping stone, a ladder, if you like, of engagements which we can develop in a reasonable way, where we can demonstrate that what the product does, now every doctor will tell you, if you're not a doctor yourself, I'll tell you, is looking for safety. Safety is all about toxicity, and therefore making sure that whatever you do isn't going to poison the patient. And it's also worth saying that one of the most sensitive organs in the body to toxicity and damage by reactive oxygen species in particular is the kidney. So the first thing we've got to do is to have a treatment which can be shown to be safe in use in the kidneys. If it then does the job that it does in nature, which is to take up the reactive oxygen species, stop the oxidative stress, which is so damaging to kidneys, then we're in a very good position. So clinically, would we want to go and try and do that first in women who are carrying a baby? Maybe not the best choice. A nice place to do it would be a group of patients who you know are going to actually receive a particular insult to their kidney in a metered dose, so you know how much is going in, and therefore you know how much can be used of your product to balance that. So our program is to go in, look quickly at patients who are going to receive a single acute dose, as it's called, a single dose, and then we can move on into each one of the treatments in PRRT, which you've seen, the radionuclide therapy, where you're giving a metered dose on a known day to a patient who is going to receive that therapy anyway. And we can hopefully show in a very short period of time that those patients will benefit because their kidneys will not be negatively affected by the radioactivity as they would normally have to suffer. If we can do that, we have this proof of concept, and a proof of concept of the mechanism of action. Because if you can do one reactive oxygen species, the physics and the physiology are the same for all reactive oxygen species. Not the same for toxins, those are different things. Reactive oxygen species are electrically charged. If you can take out that electrical charge, it makes the product. The product of the two things together, the reactive oxygen species and the Rosgard, metabolized out of the body, eliminated safely.
Do you happen to already have a ballpark estimate of how much of Rosgard you can put in without creating damage?
Yes, we can have. Within the toxicity studies that have been conducted so far, the doses which we've used across several different animal species have been consistently shown to be effective. When you take that effective dose and look at how much you need to put in there before you notice any damage at all, it's about 100-fold. So 100 times the dose that we would expect to work well in the animals.
Wow, that seems pretty safe.
It's remarkable again, and maybe nature tells us and teaches us a lot of things how to do them right. When you're developing a drug, and I've, as you know, been involved in many of those, the really difficult thing is to find a drug which is effective in doing what you want at a dose which is not toxic. And this window of opportunity, the therapeutic index, the therapeutic window between the two, is really determinant of whether you can actually treat people effectively without putting them at risk. Here, we look like that window of opportunity is big, and therefore we're going to have a good opportunity to treat patients well.
I mean, it is a good thing that you can treat possibly preeclampsia and kidney failure. But for an investor, the real important thing here is what you're worth. And it's a question of the addressable market times the likelihood of success. And what you pay for it. So your CEO earlier talked about one possibly addressable market of $8 billion but what might your market share of those kind of treatments be? And then you have preeclampsia, which is pretty much a wild card.
Well, the answer to that question actually lies back in my history to some extent. Because when we started treating cancer at Roche, all that were available were the standard chemotherapies, and they were a small market. We could not get the people in the company really excited about it until we licensed in a product called Herceptin, and that was a biologic. Now, you might say, what's this got to do with A1M? Well, that biologic came in and opened up the whole monoclonal antibody field in the treatment of cancer, which, as we all know now, is worth many, 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 many billions of dollars. The treatment of kidney disease, particularly attacks from reactive oxygen species, is in its infancy. There's nothing out there. All the drugs which have been tried have a very narrow window of opportunity. What they do is actually very close to being having toxic effects of their own before they can do anything worthwhile for the kidney. This is the opportunity which I've come to join in. Basically, what we have is another turning point in medical history, I believe, here. And with that, what's the value? Well, those numbers, the ones which we've got up there, are just looking at if we treated a small number of patients successfully with what they call with PRRT for one kind of tumours, and you multiply the number of people that received that, and you say, we could probably give them twice as much. That's the number you're looking at there. Those tumours are called neuroendocrine tumours. They're quite rare. People are now developing PRRT for prostate cancer, which is one of the most prevalent of the cancers that affect people. If PRRT is used for prostate cancer, multiply that by a few more billion.
And I'm sure you're hoping for 100% market share in that case, but that
won't
happen.
We're not treating cancer. What we're doing is we're making the treatment of cancer safer. And that's a totally different proposition. Again, I can go back to my history and look at bringing anti-nauseants into the treatment of cancer because anti-nauseants were needed in order to allow patients to have the cytotoxics, which were so nauseating. Many, many people could not tolerate their therapy. When the anti-nauseants came in, the market was open much more widely. Radiotherapeutics at the moment are an excellent field. Anything which has a bound peptide which can go and find the tumour which is somewhere in your body is an excellent modality for being able to treat cancer which has spread. You can't use surgery, you can't use beam radiation therapy because the patient would suffer terribly. If you can go and chase these things down and kill them selectively, that's a great modality. If we can make that safer for patients, then we've got a huge opportunity across all cancers sooner or later.
Okay, as a final question, then you're free to speak your word. We're talking about a multi-billion-dollar industry, and you seem to have one more or less unique pathway into this. And at the same time, you're doing this share issue at a valuation of $25 million. How did you and the board come up with the relation between that market opportunity and the valuation of the company?
The market opportunity is clearly going to be something which will take many, many years to bring about. It does. The industry as such is there to help us do that. When you look at the current cost of bringing a major drug, which this is likely to be, to market, it's somewhere between $1.2 and $1.4 billion. Our contribution to that clearly has to be as significant as we can make it. But right now, what we're talking about is less than 1% of the development cost of the drug that we intend to bring to market. It tells you that this is not a cheap game. If you get into it, you need to be in, you need to follow it through. What we've decided to do is to get sufficient money to do a really important thing, which is to bring quality into this. I ran into the CEO of Roche the other day at the airport, because I still live in the same town. And he said, we're sick to death. You remember it, Martin, all those years, people bringing forward great ideas, super stuff, but underfunded. Didn't do the experiments. And when we bring it forward, we want to convert it, and we've got to go back and do all of the clinical studies again. I don't want to do that to them or to whoever happens to be our partner in the future. I want us to bring quality product forward with the answers to the questions that I know you have to answer. So that's why we came up with that number, because that is going to give us the opportunity to move into discussions with partners out there who are going to be able to take that baton on into the next round and bring the hundreds of millions of dollars to bear to do the studies which will be required by the FDA and the MEA, which need to be done to bring this product to market. It's all very well us inventing something, but if we don't follow the pathway, if we can't bring it to patients, we can't take it to people who have those enormous amounts of money needed, then we won't do the job properly.
And your most likely exit is also to license away the patent rights, or even sell the entire company. That could happen pretty quickly, I guess, regarding the timeline.
It's quite possible. Once people see that we have the data, and this is why the clinical plan is accelerated, once they see that we have the data for the proof of concept of the mechanism of action, I'm already having conversations with people saying, that's really interesting. If you can do oxidative stress, if you can do kidneys, bring us the data. So your answer is right. But I think partnering away is not the way I look at it. It's bringing in royalties and milestones and value to the company. Somewhat different. If somebody wants to buy, well, we can't stop them.
All right. Thank you so much. That's all I had.
My pleasure.
Thank you, Martin.
Thank you.
Thank you very much.
Thank you.
Thank you.
Thank you.